So we move now on to the citric acid cycle. I don't know if we'll spend 3 or 4 lectures on this topic, but we're going to treat it very much the same way we treated our glycolysis chapter. We're going to learn about the process from a very carbohydrate-centered standpoint. We're going to learn about all the enzymes of the process. We'll do a very similar activity like we did with glycolysis, where we're going to line up all the structures. We'll learn about how this pathway is regulated, and then we'll talk about things that kind of come in and out of this pathway. So as I mentioned, we're going to learn about this pathway, and we're going to be very carbohydrate centered as we learn about the 3 main kind of processes in the heart of metabolism, which are glycolysis, the citric acid cycle, and then electron transport and oxidative phosphorylation. We're going to learn it from a carbohydrate centered standpoint because your body runs on glucose, and so that's an important component to understanding this process. So we learned about glycolysis, then we learned about sources of glucose in terms of glycogen metabolism and gluconeogenesis, and now we're kind of thinking about, well, if we want to be more efficient in our fuel use, we need to be able to squeeze all of the energy out of that fuel. We need to fully oxidize the fuel, which means we need to have other molecules that help harness that fuel in terms of electrons. So we've talked about NADH and FADH2. We're not going to learn about some of these alternative energy sources, but I do want you to just kind of remind you that there are other organisms that use other electron acceptors. We know our final electron acceptor to be oxygen. So this is a nice little figure that we have here, just as an overview to remind us that, again, we're very carbohydrate centered as we learn about glycolysis, we learn about the citric acid cycle, and again, down here, it's not shown, the fate of those reduced cofactors, NADH and FADH2, is to be regenerated under aerobic conditions in electron transport and oxidative phosphorylation. But why I like this figure is it's an important point to remind us that once we consider the oxidation of pyruvate to acetyl-CoA, we get to a branch point intermediate that is ubiquitous to any fuel type, whether it's fatty acids, amino acids, or carbohydrates. We all have those fuels end up as acetyl-CoA. And at the end of the day, big picture, what we do with acetyl-CoA depends on whether we're need state or fed state. If we're need state in terms of need energy, we continue through the citric acid cycle, and then we actually take and those reduced cofactors go on to electron transport chain and oxidative phosphorylation. However, if we are in a fed state, whether that fuel came from proteins, from carbohydrates, or from lipids, if we are in a fed state and we don't need more ATP, what happens is this acetyl-CoA gets made back into fatty acids. And I wish they had a double arrow here to remind us that acetyl-CoA to fatty acids is a reversible process. Any excess fuel must get made into acetyl-CoA and then made back into fatty acids. You remember, we learned about glycolysis thinking about it as a fed state pathway. And again, the reason is that if we've got excess fuel, we need to go through glycolysis to get to acetyl-CoA to eventually store that in the most efficient manner, which is to store as fatty acids. So again, that's a big picture thing, learning about a new important branch point metabolite as acetyl-CoA, even more important than G6P because it involves all 3 of our main fuel types. So we're going to learn about some details of the citric acid cycle. Again, some details we've already learned about that I want you to remember is it's 8 steps. It is mitochondrial. We consider it to be anaerobic. Let me flip to this next slide here. We consider it to be anaerobic because we don't see oxygen, but an important distinction, we don't allow the citric acid cycle to occur unless we're in an aerobic environment. So that's an important distinction to make. Flipping back just to this slide, which just kind of reminds us of the different metabolic intermediates and the different fuel molecules that we make. Again, NADH place in 3 spots, FADH2 in 1 spot, a single GTP in this reaction, and again, we have CO2 coming off here. So big picture, what I want you to think about with the citric acid cycle, much like we thought about with enzyme active sites, there's kind of a first half and a second half. The first half of the citric acid cycle, its purpose is to liberate the CO2 and then generate some NADH. And we end up kind of at the end of this first half with a molecule called succinate. Now what we need to do in the second half is remake succinate into oxaloacetate, because oxaloacetate is necessary to continue this cycle. So we'll talk about those details in our activity, but that's kind of the big picture component to think about with the citric acid cycle, kind of a 12 to 6 and then a 6 to 12 kind of component. The first half liberates CO2, the second half regenerates our important oxaloacetate intermediate. So two main things that we're going to get at the meat of in our in-class discussion on details and then our activity are thinking about what we call reaction 0, which is the pyruvate dehydrogenase reaction. One of the things to start kind of maybe keep track of, there's lots of things that we're going to talk about doing with pyruvate. In gluconeogenesis, we talked about pyruvate carboxylase as doing a carboxylation reaction. Now we've got pyruvate dehydrogenase going through a redox reaction to generate acetyl-CoA and NADH. Nothing that we do, but we learned about pyruvate decarboxylase when we learned about alcoholic fermentation. So lots of things with pyruvate that have pyruvate as part of the enzyme name, but something different that it does. So this is a good point maybe, and we'll have this as part of our activity to review the different things that we can do with pyruvate. This enzyme complex, pyruvate dehydrogenase is an enzyme complex. All these will look like little pieces of gumballs down here, really represent different individual enzymes. There's 3 kinds of enzymes that are in this complex. There's lots of copies of each of those. So we'll actually, in a role-playing activity, learn about how these all work together to do this fundamentally simple reaction, which is remove a carbon atom from pyruvate, making acetyl-CoA, and then producing some more NADH. So again, details here with these different kinds of enzyme active sites we'll get to in class. Again, we'll role play kind of this transfer of the acetyl group through these different complexes. And then we'll kind of talk about the different important prosthetics groups and how many of them are vitamin B derivatives. So again, the last thing we'll spend some time doing, just like we did with glycolysis, we're going to put out all of the structures for glycolysis. Let me finish getting through. So again, another kind of important component. Here's kind of the structure of the pyruvate dehydrogenase multi-enzyme complex. All right. So the last thing we'll do in our in-class activity is we'll throw out all the structures for the citric acid cycle. We have to set up our play space a little bit different because it's not like we have one table that's stage 1 and one that's stage 2. We're actually going to use one of the larger lab benches so that we can set these up in a circle, right? Because it's a cyclic process. So 10 structural players, we're going to include the pyruvate dehydrogenase reaction. And we're going to try to put these structures in order, assign enzymes, and part of your in-class activity and one that will continue into next class is thinking about mechanisms for some of these processes and then thinking about assigning enzyme names, coenzymes, co-substrates, cofactors, all of that to the process. And then in the following class, we'll actually break apart and look at the individual enzymes. So that's it in our introduction to the citric acid cycle.